While you're standing in the supermarket aisle admiring those perfect uniform tomatoes, flawless skin, brilliant red, stacked like factory widgets, you're looking at a ghost. That tomato has lost 40% of its calcium since 1950. The broccoli in your cart. You'd need 10 of those heads to match the nutrient density of a single one from 70 years ago. The corn tortilla in the refrigerated section will dissolve into mush if you leave it in water for an hour. Meanwhile, the vegetables grown by Haudenosaunee farmers in the 1400s were so nutritionally dense, so chemically sophisticated, that a European who stole the seeds, but not the processing methods, triggered epidemics that killed thousands. Modern agriculture didn't improve on indigenous crops. It hollowed them out, turned them into biological balloons filled with sugar and water, and called it progress. Walk through any American grocery store and the produce section looks like a triumph. The tomatoes are tennis ball round. The apples shine like they've been waxed. The ears of corn are identical in length, each kernel a perfect yellow pearl. Everything is big, clean, and blemish-free. This is what we were taught food is supposed to look like. But in 2004, a biochemist named Donald Davis at the University of Texas published a study that should have been front-page news. He analyzed USDA nutrient data for 43 crops, comparing their mineral and vitamin content in 1950 to their content in 1999. What he found was a systematic collapse. Protein had dropped by up to 38%. Calcium, iron, and essential vitamins followed the same trajectory, declines of 15 to 40% across the board. This wasn't a blip, it was a pattern across nearly every vegetable and grain tested. The plants were growing faster, getting bigger, but the chemistry inside them was thinning, out like paint diluted with water. Davis called it the dilution effect. Modern plant breeding focuses on 3 targets: maximize yield per acre, increase pest resistance, and extend shelf life. Nutrient density is not on that list. The result is that a fast-growing plant accumulates carbohydrates, sugars and starches, faster than it can pull minerals out of the soil. You get a vegetable that weighs more, stores longer, and ships better, but it's mostly water and sugar. The micronutrients that made it food, the iron, the calcium, the vitamins, are present in trace amounts, like an echo of what used to be there. A modern tomato looks like a tomato. It has the right color, the right shape, but chemically, it's a shadow. You are eating the idea of a vegetable. Before Europeans arrived in the Americas, Haudenosaunee and other indigenous peoples across the continent weren't just planting vegetables. They were running biological systems. The 3 sisters, corn, beans, and squash, gets taught in elementary school as a nice story about cooperation. That's a disservice. This was agronomic architecture. Start with corn. Botanically, it's Zea mays, and it uses a photosynthetic pathway called C4. Most crops, wheat, rice, soybeans, spinach, use C3 photosynthesis, where the enzyme rubisco sometimes wastes energy grabbing oxygen instead of carbon dioxide. Corn uses C4 photosynthesis. It has specialized cell structures that concentrate CO2 around rubisco, eliminating that error. The result is that corn acts like a high-efficiency solar panel. It converts sunlight into chemical energy faster than almost any other crop. But corn's role in the system isn't just chemical, it's structural. It grows tall and vertical with narrow, upright leaves. This creates a scaffold for the beans to climb without shading the squash below. The root system is massive and fibrous, stabilizing the soil mound and preventing erosion during heavy rains. It's the skeleton of the system. Then you have the beans, Phaseolus vulgaris. These are the system's on-site fertilizer factory. Modern industrial farming requires synthetic nitrogen fertilizer, which is produced through the Haber-Bosch process. That process uses enormous amounts of energy, between 1 and 2% of the entire world's energy budget, to force atmospheric nitrogen into a form plants can absorb. Beans do this for free. They don't do it alone. The bean plant secretes chemical signals called flavonoids into the soil. These signals attract a specific type of bacteria, Rhizobium, which infects the bean's roots. In response, the plant builds specialized organs called root nodules. Inside these anaerobic nodules, no oxygen, The bacteria convert atmospheric nitrogen gas into ammonia, which the plant can use. The bacteria get fed sugars from the bean's photosynthesis. The bean gets nitrogen. When the bean plant dies or its roots decay, that nitrogen gets released into the soil, fertilizing the corn and squash for the next growing season. It's a slow-release fertilizer system timed to the needs of the soil. Squash, cucurbita, covers the ground. Its broad leaves act as living mulch, shading the soil and reducing water evaporation by up to 60%. In arid regions like the American Southwest, where these systems were refined over centuries, water retention was the difference between crop success and failure. The shade also keeps the soil cool, protecting the microbial communities, the bacteria and fungi that would be sterilized by direct UV radiation and heat. But squash isn't just passive, it's chemically aggressive. Many squash varieties exhibit allelopathy, which means they release biochemicals from their roots or decaying leaves that inhibit the germination of competing plants. It's botanical warfare against weeds. The stems are covered in calcified spines, rigid, sharp trichomes that act as a physical barrier against small mammals and insects trying to climb up the corn stalks to get at the ears. Some indigenous farmers also planted a fourth component, Rocky Mountain bee plant, Cleome cerulata. Its job was to attract pollinators, bees and wasps, to ensure the beans and squash were fertilized. Corn is wind-pollinated, but the others aren't. Without the bee plant, yields dropped. This wasn't gardening, it was systems engineering. Each organism created the environmental conditions the others needed to survive. In the 1700s and 1800s, a disease called pellagra swept through parts of Europe and the American South. The symptoms were horrific: severe dermatitis that made the skin crack and peel, chronic diarrhea, neurological degeneration that led to dementia, and eventually death. Doctors called it the disease of the 4 Ds: dermatitis, diarrhea, dementia, death. The cause was corn. Or rather, it was corn without the chemistry. When Europeans took maize back to the Old World, they took the seeds. They didn't take the instructions. Indigenous peoples across Mesoamerica and North America had developed a processing technique called nixtamalization. The word comes from Nahuatl, nixtli, ashes, and tamali, dough. The process involved boiling corn kernels in water mixed with wood ash or slaked lime, calcium hydroxide. On the surface, it looks like a simple cooking step. It's not. It's alkaline hydrolysis, a chemical transformation. Raw corn contains niacin, vitamin B3, but it's chemically bound to hemicellulose, a complex carbohydrate that human digestive enzymes can't break down. That niacin is locked away, biologically useless. When you boil corn in an alkaline solution with a high pH, the hemicellulose bonds break. The niacin is released and becomes 100% bioavailable. Your body can now absorb it, but that's not all the process does. The alkaline treatment also improves the availability of 2 essential amino acids, lysine and tryptophan, which are present in corn but difficult to access in their raw state. And it destroys up to 97% of aflatoxins, toxic compounds produced by mold that grows on stored grain. Aflatoxins are carcinogenic and can cause acute liver damage. Nixtamalization made corn safe to store for years without refrigeration. Europeans didn't know any of this. They planted corn, harvested it, ground it into flour, and ate it. And then pellagra appeared. It ravaged Italy. It killed people in Spain. In the American South, where poor populations relied heavily on cornmeal, it became epidemic. Thousands died. Indigenous farmers had been performing chemical engineering that modern science wouldn't formally document until 1974, when a researcher named Solomon Katz published a paper in Science explaining the biochemistry of nixtamalization. By then, indigenous peoples had been doing it for at least 3,000 years. The Europeans thought they were taking advanced technology when they stole the seeds. They took the shape of the technology and left the knowledge behind. Here's a problem. Corn is an incomplete protein. It's high in an amino acid called methionine, but it's almost completely deficient in lysine and tryptophan. If you try to survive on a diet of just corn, you will develop protein malnutrition. Your body can't synthesize those missing amino acids on its own. They're essential, meaning you have to get them from food. Beans have the opposite profile. They're rich in lysine and tryptophan, but low in methionine. Indigenous peoples didn't eat corn alone. They didn't eat beans alone. They ate them together, often in the same dish like succotash. When you combine corn and beans, the amino acid profiles lock together like puzzle pieces. The methionine from the corn fills the gap in the beans. The lysine and tryptophan from the beans fill the gap in the corn. The result is a complete protein with a bioavailability profile that rivals beef. This wasn't an accident. It wasn't a cultural preference that happened to work out. It was a biochemical necessity encoded into cuisine. Indigenous societies across the Americas, Mesoamerica, the Southwest, the Eastern Woodlands, independently developed food pairings that ensured protein sufficiency in a largely vegetarian diet. Modern industrial monoculture breaks this. Corn is grown in one region, shipped thousands of miles, and eaten in isolation. Beans are grown somewhere else. The synergy is lost. You end up needing to eat animal protein to compensate, or you take synthetic supplements. The biological technology that solved the problem for free is abandoned in favor of a system that requires constant external inputs. Modern nutrition labels measure food in calories. Open any packaged product and the first number you see is the caloric content, how much energy the food contains when burned. This has trained us to think of food as fuel, nothing more. A calorie of corn syrup is treated as equivalent to a calorie of nixtamalized corn. A calorie of white bread is the same as a calorie of bean stew. By that metric, a can of soda and a bowl of Three Sisters succotash are functionally identical as long as they contain the same number of calories. This is a catastrophic oversimplification. Calories measure energy, but they don't measure information. They don't account for the micronutrients, the vitamins, minerals, and phytochemicals that regulate every metabolic process in your body. They don't measure fiber, which controls blood sugar and feeds gut bacteria. They don't measure the bioavailability of protein or the presence of anti-inflammatory compounds. A high-calorie food can be nutritionally barren. A lower-calorie food can be biochemically rich. The Three Sisters system didn't just produce calories. It produced a complete nutritional matrix. The corn provided complex carbohydrates and energy. The beans provided protein with a full amino acid profile. The squash provided beta-carotene, potassium, and fiber. When eaten together, often with wild greens, and small amounts of game, the diet covered every essential nutrient a human body requires. Modern industrial crops are bred to maximize caloric yield. Corn has been selected for high starch content. Wheat has been bred for gluten strength to improve baking. Potatoes have been optimized for size and storage. But the secondary compounds, the polyphenols, the flavonoids, the trace minerals, have been inadvertently bred out. These compounds don't contribute to yield. They don't improve shelf life, so they disappear. The result is that modern diets are simultaneously high in calories and low in nutrition. You can be overfed and undernourished at the same time. Indigenous diets, by contrast, were calorically modest but nutritionally dense. Every bite carried information your body could use. Not just energy to burn, but the chemical building blocks for cellular repair, immune function, and metabolic regulation. We stopped measuring food quality and started measuring food quantity. That shift alone explains much of the modern health crisis. Modern agriculture treats soil like a passive substrate, a medium to hold plants upright while you dump fertilizer on it. Indigenous agriculture understood soil as a living system. The difference shows up in the roots. In a modern industrial cornfield, the roots are shallow. They spread out horizontally in the top 6 to 12 inches of soil where the synthetic fertilizers are concentrated. Below that, the soil is often compacted by heavy machinery. It's dense, oxygen-poor, and biologically dead. If you cut a cross-section, you'd see a thin layer of active root hair, then nothing. The plant is functionally rootless below the plow line. In a traditional milpa, the roots go deep. Corn roots in ancient landraces can extend down several feet, anchored by a fibrous network that pulls water and nutrients from far below the surface. Bean roots penetrate even deeper, their nodules scattered throughout the profile like biological factories. Squash taproots dive down to access moisture, reserves. If you cut a cross-section of that soil, you'd see a three-dimensional web of roots, fungal hyphae, and microbial colonies. It's porous, dark, and alive. The Hopi people in what is now Arizona farm corn in a high desert that receives less than 10 inches of rain per year. Modern agronomic textbooks say corn requires 20 to 30 inches. The Hopi have been doing it successfully for over 1,000 years. The mechanism is soil physics. Standard farming practices plant corn seeds 1 to 2 inches deep. Hopi farmers use a planting stick made from greasewood to punch a hole 6 to 18 inches deep. They drop the seed into the bottom of that hole, down into what hydrologists call the capillary fringe, the zone where moisture from the water table is drawn upward by capillary action. The seed germinates in contact with that moisture. The specialized root structure of Hopi corn, called the mesocotyl, elongates drastically to push the shoot up to the surface. By the time the plant emerges, it's already anchored to a stable water source that surface-planted corn would never reach. They also plant in clumps. Instead of single seeds spaced out in rows, Hopi farmers drop 10 to 20 seeds into one hole. Most of those plants will die. The outer ones wither first, creating a windbreak that protects the inner plants from the scouring desert sand. The dense cluster of leaves traps humidity, creating a microclimate at ground level where evaporation is slowed. It's a living sacrifice system. Some plants die, so others thrive. In the Amazon, indigenous cultures faced the opposite problem. Tropical rainforest soil is terrible for agriculture. It's acidic, nutrient-poor, and any fertility gets washed away by constant rain. Modern farmers who clear rainforest for cropland find that the soil is exhausted within 3 to 5 years. But there are patches of soil in the Amazon called terra preta, black earth. These soils were engineered by indigenous peoples around 2,000 years ago. They mixed charcoal, bone, manure, and broken pottery into the soil. The charcoal, biochar, has a porous structure with a surface area of hundreds of square meters per gram. It acts like a coral reef for soil bacteria and fungi, and it holds nutrients chemically, preventing them from leaching out during rain. Those soils are still fertile today. You can dig up terra preta and plant crops in it without adding fertilizer, and they'll grow. The soil was engineered to last longer than the civilizations that built it. Modern cleared rainforest goes barren in 5 years. Indigenous engineered soil has been productive for 2,000 years. 99% of the corn grown in the United States is genetically similar. It comes from a narrow pool of hybrid varieties bred for uniformity, high yield, and compatibility with industrial equipment. This is efficient in the short term. It's catastrophic in the long term. If a pathogen, a fungus, a virus, a bacterium, evolves to exploit a weakness in that genetic line, it can spread across millions of acres without encountering resistance. There's no genetic diversity to slow it down. This is happening right now with bananas. 99% of bananas sold globally are a single variety called the Cavendish. A fungal disease called Panama disease tropical race 4 is spreading through plantations, and there's no resistant strain ready to replace it. The global banana supply is collapsing in real time. Indigenous farmers never created genetic monocultures. A single Hopi cornfield contains more genetic diversity than the entire corn belt of Iowa. Farmers saved seeds from the plants that performed best in their specific environment, those that survived the drought, resisted the local pests, or produced the most in poor soil. Over generations, this created thousands of distinct land races, each adapted to a microclimate. That diversity was a distributed genetic library. If one variety failed, others survived. Modern agriculture optimized for control and predictability. It traded resilience for uniformity. The result is a food system that is one pathogen away from collapse. Glyphosate is the most widely used herbicide in the world. It's marketed under the brand name Roundup. Genetically modified crops called Roundup Ready are engineered to survive being sprayed with it so farmers can kill weeds without harming the crop. On paper, it's elegant. In practice, it's destroying the soil. Glyphosate doesn't just kill plants, it functions as an antibiotic. It disrupts the shikimate pathway, a metabolic process used by plants, fungi, and bacteria, but not animals. When you saturate soil with glyphosate, you kill the rhizobium bacteria that fix nitrogen. You kill the mycorrhizal fungi that extend plant roots and help them absorb phosphorus. You sterilize the microbial ecosystem that makes soil alive. Once the soil biology is dead, the plants can't access nutrients naturally. They become dependent on synthetic NPK fertilizers, nitrogen, phosphorus, potassium, applied in chemical form. But plants grown in dead soil are weak. They lack the secondary metabolites and structural compounds that come from a healthy root-fungal partnership. Weak plants attract pests, so farmers add pesticides. The pesticides kill beneficial insects along with the harmful ones, further destabilizing the ecosystem. The cycle compounds. Every input creates the need for the next input. It's agricultural addiction. The milpa system was the opposite. Beans fed nitrogen into the soil. Squash suppressed weeds through allelopathy and shading. The diversity of crops supported a diversity of insects, so no single pest could dominate. The system regenerated itself. Each season, the soil got richer, the microbial networks denser, the plants healthier. Modern monoculture extracts. Indigenous polyculture regenerates. We chose the first because it looked more productive in the short term. We're now learning what it costs. That supermarket tomato in your hand weighs more than a 1950s tomato. It's redder. It has no bruises, no blemishes, no irregularities. It will sit in your refrigerator for 2 weeks without rotting. But it has lost 40% of its calcium. It has lost 15% of its iron. It has lost 38% of its riboflavin. If you were to chemically analyze it next to its ancestor, you'd find that the modern tomato is mostly water and fructose. The minerals and vitamins that made it food, the compounds your body needs to build bones, transport oxygen, generate energy, are present in trace amounts. You are eating a ghost. It looks like a tomato. It has the shape, the color, the texture. But the chemistry that made it nutritious has been bred out in favor of shelf life, uniformity, and yield. The same is true for the broccoli, the corn, the apples. The nutrient losses documented by Davis mean you'd need to eat significantly more modern vegetables to match the nutritional value of their predecessors. The food hasn't just gotten less nutritious, it's been diluted to the point where it barely qualifies as food. Meanwhile, the crops developed by indigenous farmers, corn bred for deep roots and drought tolerance, beans engineered to self-fertilize through bacterial symbiosis, squash that controls its own microclimate, were chemically dense, nutritionally complete, and regenerative. They didn't require external inputs. They didn't deplete the soil. They didn't create dependency loops. Modern agriculture treats plants as factory products. You input chemicals, you output calories. Efficiency is measured in yield per acre, cost per bushel, shelf life in days. Nutrition is not part of the equation. Indigenous agriculture treated plants as nodes in a living network. You input biology, bacteria, fungi, genetic diversity, and you output resilience. Efficiency was measured in how little external labor and resources the system required, how well it survived, drought or pest outbreaks, how long the soil stayed fertile. The Three Sisters was perfected by the year 1491. It was a closed-loop biological machine that could feed populations for generations without degrading the land. Everything we've done since, hybrid seeds, synthetic fertilizers, glyphosate, genetic modification, has been a trade. We traded nutrient density for appearance. We traded resilience for control. We traded partnership with biology for chemical dependency. And we called it progress. The most nutritionally advanced crops on Earth were already in the ground 500 years ago. We just decided not to learn from them.